Good morning friends, myself Dr. Ashish Panchal from Trisha Institute of Biology. In last video, we already discussed the development in plant and development of the plant required some plant growth regulators. But from where this plant growth regulator were discovered? So here we will discuss the discovery of PGR, means the discovery of the plant growth regulator. Actually, the discovery of the plant growth regulator have been accidental, means they are not actually discovered, they are discovered by accidentally. The first time the Charles Darwin and his son Francis Darwin observed the coleoptile of the canary grass plant. Now, what is coleoptile? Coleoptile is the part of plumule which protect the plumules, as we discuss in the monocot seed. So, in monocot seed, we know that it is endospermic part and this is the embryonic part, and they possess the plumules and radicle. This plumules is protected by the coleoptile. So, the Charles Darwin and his son Francis Darwin was observed the coleoptile of the canary grass, and this canary grass respond towards the light. When they illuminate the unilateral light on the coleoptile of the tip of the canary grass plant, then he observed that the tip of the coleoptile bend towards the light, and that process is known as the phototropism. That process is known as the phototropism. And the series of experiment conclude that a tip of the coleoptile has some chemical which is responsible for the phototropism. Clear? So, first time Charles Darwin and his son Francis Darwin observed that the effect of light on the coleoptile of the canary grass plant. And what he observed? They observed that the tip of the coleoptile is bent towards the phototropic, means towards the light, and that process is known as the phototropism. Then after the auxins, the first time isolated by F.W. went from the tip of the oat coleoptile. Okay, here this is the figure. When the light illuminate on the tip of the coleoptile bend towards the light, that is known as the phototropism. Okay, now next is the discovery of zebralic acid. So the bacane actually foolish seedling, it is the disease which is found in the rice seedling. Okay, and it disease caused by the fungus known as the Zebralic fujikuroi. This Zebralic fujikuroi is the fungus which is responsible for the bacane disease. The E. kurosova, it's a name of scientist, reported that the appearance of the symptoms of disease in the healthy plant of the rice when it was infected by the sterile fungi. Which fungi? So this is Zebralic fujikuroi. So what exactly Kurosova did? So Kurosova reported that the appearance of the symptoms of the disease in the healthy plant and the rice plant when it is affected by the sterile fungi. Then after, he said that the substance which is present in the fungus is known as the zebralic acid. Okay, so this is the discovery of the zebralic acid. Now next, the Abs Cook and his worker studied on the internodal segment of the tobacco stem, and he prepared the callus culture. Now callus means undifferentiated mass of cell, means it is a group of cell which is not differentiated. If they are differentiated, they converted into tissue system and organ, but here they prepare the mass of undifferentiated cell, it is known as the callus. And how they prepare this callus? So they prepare the callus with the help of the auxins, and in addition to auxins, they added the vascular tissue extract, yeast extract, and coconut milk and the DNA. Then after, Skoog and the Miller discovered the crystalline substance which is present in this extract, and they gave named as a kinetin. Means this crystalline substance is called the kinetin, which is responsible for the cytokinesis, and we know that the cytokinesis, it's a breakdown of the cell membrane or the division of the cell membrane during the cell division. Okay, then after, in mid 1960s, the three scientists reported the three different chemical under the name of inhibitor B, abscessin 2 and the dormin. This inhibitor B, abscessin 2 and the dormin, these three chemicals, the compound are chemically identical, means chemically they all are similar. So then after, they all put under the one name, and it is known as the abscisic acid. Then at last, the cousin reported that the ripened orange released some volatile substance, and this volatile substance is known as the ethylene, which is responsible for ripening the other fruit. Means this ripened orange released the volatile substance, and this volatile substance has done the ripening of the unripe banana, and that volatile substance is known as the ethylene. Okay, so this is all about the discovery of the various hormone. Look at the discovery of the plant growth hormone. The first discovery of the each of these 5 hormones are the accidental. They are not discovered by experimentally, they are discovered by the accidentally. Charles Darwin and his son Francis Darwin observed that the coleoptile of the canary grass bend towards the light when they illuminate the unilateral light or unilateral illumination on the tip of the coleoptile. Then series of experiment conclude that the tip of the coleoptile is bent towards the light, and that process is known as the phototropism. Okay, and it will be explained in this figure. Auxins first time was isolated by F.W. Wendt from the tip of the oat coleoptile. Then the discovery of the zebralic acid. Actually, the zebralic acid, okay, the bacane means is the foliage seedling, and it is found in the rice seedling. It is the disease. It is found in the rice seedling, and the disease is caused by the fungus known as the Zebrella fusicuroi. The E. Kurosawa reported that the appearance of the symptoms of disease in the uninfected or the healthy rice seedling when it is infected by the sterile fungi. And then after, he was reported this substance as a zebralic acid. Then the discovery of the cytokinins or the discovery of the kinetins. Epskog and his workers studied in the internodal segment of the tobacco stem, and they prepared the callus of tobacco cells. Okay, and this callus is known as the undifferentiated mass of cell. How they prepare this callus? So they use the auxins with vascular tissue extract, yeast extract, coconut milk extract, and the DNA. And then after, the Skog and the Miller isolate the crystalline substance from this extract, and it is known as the kinetin. This kinetin is responsible for the cytokinesis, means it is responsible for the division of the cell membrane, and it is help in the formation of the callus. Then during the mid 1960s, the 3 scientists reported the 3 different substances, inhibitor B, abscessin 2, and the dormin. But later on, this all 3 substances chemically they all are identical, means chemically they all substances are similar. Okay, so they put under the one name that is known as the abscisic acid. Then cousin reported that the ripened orange released the volatile substance, and this volatile substance is known as the ethylene, which hastening the ripening of the unripe banana. So the ripened fruit release some volatile chemical, it is known as the ethylene, and ethylene is the gaseous form of the hormone. Clear? So this is all about the discovery of the plant growth regulator. In next video we will discuss the each and every hormone in detail. Okay, thank you.